Hi folks, it's Gene here. I'm in Spring, Texas, and today we're going to talk about properly collecting a soil sample. Okay, so our subject plant is going to be this pineapple guava here, Acacelloiana. Some of you may know it as Feijoa celloiana. You can see that it's in really rough shape. And then one of the reasons that we want to try to turn it around is because all these others here, over here on the west side of the property, are doing so well. They're really thick and dense and dark green. They have a ton of brand new foliage, and they have a lot of flowers as well. So the first thing we're going to do is we're going to get our soil sample bag out, and we're going to write the name here, and we're going to put pineapple guava. The lab that I'm going to send this sample to may have a baseline of some of the pineapple guava's preferences, like pH. So it's important to put down, which plant you're going to do. So we're going to start with that. We're going to put our gloves on, and we're going to talk about the three primary tools that we use to collect a soil sample, and I'll tell you which one is my favorite and why. The most common tool is the soil knife. It's not my favorite. And you would just dig here and collect a sample, right? And so, as you can see as I'm digging, in a lot of ways I'm cultivating more than digging, right? I'm not really defining a clear layer. So I've kind of mixed the first couple inches with the next couple of inches. And so if I have a soil that has the first 2 inches has high organic matter, which is very common in urban and suburban landscape settings, I don't want that in the bag because I don't want to get a false reading that the organic content in the soil is high. I want to test the soil a couple of inches below the surface, somewhere between 2 and 8 inches depth. That's where I want the roots to be the healthiest. That's where I want the highest cubic volume of fibrous feeder root mass. And so it doesn't help me to put the first 2 inches of soil in the bag. I'm not sampling for grass. If I was sampling for grass, it would be instructive to put the first 2 inches of soil in the bag. Okay, so the next tool is the Dutch auger. This is a Dutch auger. This is a nice 1.5 inch diameter. The one we use the most is about 2 inches in diameter. And this is pretty cool. It allows me to pull a pretty cool sample. Let me show you how we do it here. It's just like augering a hole, like a post hole digger. It's just much smaller in diameter. And one of the best advantages of this, if we're working on a really difficult site, or if we suspect contamination from industrial contamination or a gas leak, then we might have to go deeper, right, than your typical root zone. And so I'm going to break this off and lay it down. And can we zoom in on this? And I'm going to take this, it's going to be my first piece, and I'm going to take that out, and then I'm going to go a little bit farther. And I'm going to pull a little bit more. This is a great tool. Most of the arborists in my office, this is their preferred tool, right? So we pull this out, and we set it next to it. And if we put this out on a piece of paper, sometimes we can see various levels. All right? So this is a pretty good method. Let me grab my next one and we'll talk about it. This is the standard soil probe. The main reason most of the arborists in my office don't like to use this is because it's really hard to clean it out once you've collected a core. And if the ground's hard, it's really hard to get a good sample. But see, I'm pulling out a nice clean core, right? And if you look at this, we can look at that first 2 inches of dark material. That's the high organic content. And then from here down, we can see that it's much more compacted, much lower in organic content, and it's clay, right? This is the stuff I'm going to put in the bag. And those of you who have trouble cleaning this out, I mean, if you're a legit tree care professional, you know where to get an eye bolt. This is a drop forged eye bolt. This is not the kind I use in trees. This is a 3/8 inch. I don't really use this size. I use a larger one. But I accidentally got some of these delivered, and I use it. Check this out. It's great for cleaning out your probe. And then you can just store it. So that takes away the problem of keeping this particular one clean. Now I want to go to the soil bag, and I want to show you where it says fill to this line. Now I called the lab and I said, do we really need to fill to this line? She said, absolutely not. She said, the reason it says fill to this line is because people send in such crappy samples that are high in organic matter. They have compost, they have leaves, they have roots, they have wood chips mixed in. And what she does is she lets it all out on a bag, and she removes all recognizable plant parts from the sample before she does her analysis to it. So you don't need to fill to this line, as long as you're putting the right stuff into the bag, which is the stuff from 2 inches down to about 6 or 8 inches down, the stuff where you really want to see healthy, fibrous feeder roots, right? Okay, so I've got a few other places in the landscape where I could collect a sample. Up in the front, we've got some ginger that's not doing great. We've also got some Indian hawthorns that aren't doing so great up there. So it's not going to kill me to collect another sample, get another bag for a couple of other plants. But for here in the backyard, this is the plant that I'm primarily concerned about. So next, we're going to ship it off to the lab. Thank you very much.